Good afternoon, my name is Fernanda Dalarmi and I'm a master's candidate of the entomology graduate course at Ribeirão Preto, USP. Today I am pleased to present to you my research project entitled Reproductive Capacity of Frieseomelitta varia and the Notch and EGFR silent pathways. We used a native Brazilian stingless bee as a study model, but why use this species? They are eusocial bees which means to stand on three pillars. They have a division of labor with a caste system involving sterile or non-reproductive females helping those who reproduce. There is cooperation between members of the colony in caring for the offspring, and finally there is overlapping of generations capable of contributing to the colony's functioning. Another really interesting characteristic is the irreversible sterility in F-varia workers. It happens because ovarian cells undergo an intense program of cell death during the pupal stage. This results in highly modifiable and sometimes unrecognizable ovaries in adult individuals. So all these characteristics make them a great model of study to what genes are involved in reproductive capacity. In all social hymenopterans, work reproduction is limited by the physiology established during development. The Notch cellular silent pathway is highly conserved with pleiotropy roles in development, having multiple functions in oogenesis and reproduction. For example, it is responsible for controlling processes such as differentiation and specification of cell fates, and depending on the biological context, proliferation and apoptosis. This pathway has been found in bees suppressing reproduction. The EGFR silent pathway plays a key role in determining the queen and worker cases. EGFR is very important for the proper development of ovarian follicles. EGFR depletion prevents oviposition, probably due to premature death of follicular cells, playing a role in controlling cell proliferation through the interaction between EPO and NOT pathways. The main object of this project is to use different development stages, pre-imaginal and adult, of Frieseomelitta varia to investigate the participation of the Notch and EGFR pathways into the regulation of different levels of reproductive capacity of queens and workers, using as references the morphology of the ovaries and analysis of gene expression. We divided the experimental plan into three steps. The first means the selection of candidate genes, annotation of these genes, and the assembly of primers. It is expected to find all genes in the F-varia genome. In step 2, we have the sampling and production of queens, dissection of the ovaries, and the bench part. Significant differences in the expression of these genes are expected when queens and workers are compared. In the third and last step, we have immunohistochemistry microscope, where it's expected to observe specific places where these genes are acting. So far, step 1 has been completed. The 5 genes chosen from the Notch and EGFR pathway as the only representative of their pathway have been localized and annotated in the genome of F-varia. We would like to thank FAPESP and CAPES for funding the project, and also to the Laboratory of Biology and Development of Bees for all the infrastructure provided.